bacteria project, a lot of the assays I had to do were with mammalian cells. So like visualizing bacteria once they had entered mammalian cells. I did a lot of fluorescence microscopy in the spring semester of last year. And so like the mammalian cell work is pretty different than the bacterial cell work, just in terms of the time scale, the care you have to give to the cells, you know, like how, like the type of environment, like mammalian cells, you have to make sure everything's really clean, and you have to ethanol everything, get rid of the bacteria, because they will outcompete your mammalian cells if they get into your culture. Bacteria grow really fast and they're fine and happy like all the time, mostly. So it's kind of different working with bacteria and mammalian cells, but I like how I've had the opportunity to do both.
Do you think there's been any because I hear a lot about how the collaboration of IGEM is very interesting. Like, how would you compare the actual like interactions in the lab with IGEM and working in Chris's lab, or would you say they're very similar?
Okay, so I mean, as far as like collaborating with other people in Chris's lab, a lot of people in Chris's lab, actually most of them, are doing bacterial cell work. I was kind of one of the exceptions when I was working with Chris. So a lot of them are doing things that are much more similar to what we're doing with IGEM in terms of, you know, cloning parts, joining parts together, trying to make devices that do certain things. And like different people are working on making devices that do different things. But as far as the collaboration goes, there has been a little bit of crossover with parts. Actually, most of that has involved me for some reason. I'm not quite sure why. But you know, one of my parts that I was using was something that Samantha Liang, who now works in Chris's lab, had made I think a year or two ago for IGEM, and I mean, it's a part that has been used quite a bit since then. And then because I'm doing some things that are similar to Daniel in terms of, you know, like the gateway recombination reaction and kind of optimizing that, there is a little bit of crossover parts there. And yeah, so just there's a little bit of crossover parts, but I mean, as far as like the information exchange, most of it has been limited to like just common parts or common things that we're using. It's not like many of the Anderson lab people have been giving us like a lot of advice or things like that, even though most of them have been involved with IGEM at some point. Does that answer your question?
Mm-hmm. Okay. So what is your specific project with IGEM? What is your part of the whole scheme?
Okay, so I'm kind of working on a couple different things within IGEM. So there are other people working on the sound lysing device, which is like our baseline project for lysophonics. But a lot of my work has been on the gateway recombination reaction. And well, first I was testing whether we could do that in the lysate. So like our original idea was, as an application of sound lysis, we could basically mix two bacteria cultures together and have each one of them have one of the plasmids we wanted to recombine together, and also have like between these two cultures, have the bacteria producing all the reagents required for the reaction. And so we basically want to be able to mix the two cultures, sound lyse them, have them mix their contents, which would include the two plasmids we wanted to be recombined, as well as, you know, the enzymes necessary to do it. And then the reaction would proceed basically spontaneously, and then our desired product would be in that mixture, and we would be able to transform that into new set of cells and isolate the product. But so initially I was kind of testing the feasibility of doing this sort of a reaction in those conditions where you have cells that have just lysed. And just like we found out that you can do the reaction there, but the amount of enzyme or the amount of plasmid that you would need from the cells, like you would have to have too large of a volume of cells to have enough plasmid for this reaction to occur. And so we were kind of at the same time also pursuing having this reaction happen in the cytoplasm of a cell. So that's the one we're now focusing on, having the reaction happen within the cell. We have two different schemes for that right now. I'm working on making the parts for one of them, and like the particular scheme I'm working on has one of the things we want recombined, integrated into the genome, and the other thing in a plasmid that we would put into the cell that has this part integrated into the genome. And then like hopefully the recombination reaction would occur. And we're using some conditional origins of replication that only work when certain proteins are present, in order to kind of screen for the product that we want. And the other scheme that Chris is working on, and it has crossover with parts that Will Deloach made when he was here. And so that involves like having the reaction happen in cytoplasm, and it has plasmids that can be packaged and carried in phages. And so that's how we would screen for the desired product.
Because the phages would...
Well, okay, so basically once the reaction happens, so the desired product would have this particular device that would allow it to be packaged into a phage, and so it would allow it to be packaged into the phage, but like the other reactant wouldn't have that part that would allow it to be packaged into a phage. And so you could use the phages from that to reinfect new cells. And using that, a combination of that sort of screening and antibiotic screening, you would be able to isolate your desired product. So, I mean, as far as gateway in the cytoplasm, we know that we can do it. Like, we have already shown that there's a result that, like, it can work. It's just there's a lot of background right now. And so these two schemes are attempts to basically reduce the background and make sure that like we're kind of getting mostly our desired product and not the other side products that we would be able to see.
And this gateway reaction is quite important to doing synthetic biology in general.
Yeah, so the gateway reaction is, it's most commonly used for like genomic integrations and things like that. But what we use it for is kind of taking parts from you know, taking basic parts from